Good morning, good afternoon, wherever you all are in the world. My name is Samaya Vaidya, and I'm honored to present to you my project titled Therapeutic Breakthroughs DDX3 as a Novel Target in Thalassemia. Before I begin explaining the full project in detail, I'd like to give you a brief overview of what this research aimed to accomplish and how it was structured. My paper explores how computational biology and bioinformatics tools can be used to identify new drug targets for thalassemia, which is a genetic blood disorder that affects hemoglobin production. In my project, instead of focusing only on the one well known gene, which is HBB, I used a systems level approach that analyzed thousands of genes from real patient data to uncover other potentially important players in this disease. The project was carried out in four main stages: differential gene expression analysis, network and pathway analysis, target selection, and finally, virtual screening and molecular docking. Of course, I will go into depth into these much later. Overall, this paper combines data analysis, molecular biology, and computational simulations to propose a novel therapeutic direction for thalassemia, all without needing a physical laboratory. It also establishes a framework that could be applied to other diseases in the future. Okay, now I'll take you through each part of the project in more detail. So let me tell you about why we chose thalassemia. Thalassemia is a genetic disorder that's not often brought into the limelight, though it does affect hundreds of thousands of lives across India, my home country, and millions globally. Every year, around 3 to 4 lakh children are born with severe forms of this disease. That's nearly one child every minute, which, yes, is a shocking statistic, and yet one that receives far less attention than it really should. So, the problem is that thalassemia is caused by mutations in the genes responsible for hemoglobin production, as I said, most commonly the HBB gene. While that might sound very straightforward as a genetic defect, the treatment is very far from simple. Often, patients require lifelong blood transfusions, which can lead to iron overload and other complications. Though we have made advances in symptomatic treatments and gene based therapies, these options still remain very expensive and hence are inaccessible. What we noticed and what became the foundation of our research is that most studies focus solely on the HBB gene rather than looking at the broader genetic network that often influences hemoglobin regulation. What if there are other players, genes or pathways for that matter, that we're overlooking? Is it possible to find new therapeutic targets through bioinformatics and computational biology? These were the core questions that I had for my project. So, I aim to use computational biology to identify novel protein targets and potential drug leads for thalassemia by one, analyzing gene expression data, next, building protein interaction networks, then mapping distributed biological pathways, and finally using virtual screening to simulate drug binding as I didn't have access to a lab. My goal was to find a target that hadn't yet been deeply explored and to validate its potential for therapeutic development using a rigorous three-step computational framework. So, what sets this project apart? Of course, I didn't just stop at identifying differently expressed genes. I created the protein-protein interaction networks, conducted pathway analysis, and used molecular docking. Next, my dual pipeline approach, which included network and virtual screening, allowed me to discover previously unknown and underexplored protein, DDX3. Next, I also built the groundwork for future protein based treatments, which could eventually lead to more affordable and accessible therapies. So, to execute this, I combined several powerful bioinformatic tools in a logical pipeline. I started by selecting the RNA sequence dataset GSC 96060 from the GEO, from Gene Expression Omnibus, which is GEO. So this data set included samples from both and healthy individuals. Using this tool, I identified 23 significantly differentially expressed genes, which are basically the genes that you would find in a patient with thalassemia. And these were altered in patients of thalassemia versus healthy controls. We then visualized them via a volcano plot, allowing us to shortlist genes for network analysis. Next, we put these differentially expressed genes into String, which is a platform that maps protein-protein interactions. And I constructed networks with 5, 10, 20, and 50 interactors to analyze their topological features. These features were number one, the degree, which is the number of direct connections between the proteins, then between a centrality, which is basically how often a node acts as a bridge between the proteins. And lastly, the clustering coefficient, which describes how interconnected a node's neighbors are. Next, after that, we use Cytoscape to visualize and analyze these networks, identifying 5 top genes from the 23 that had originally been shown: PRKY, EIF1AY, DDX3Y, CDY2B, and BPY2. Among these, DDX3 stood out, not only because it was a hub gene, but also because it appeared in the top overrepresented pathways related to patients with thalassemia. After this, to further validate our findings, I used the Reactome pathway database to conduct pathway enrichment analysis. DDX3 consistently appeared in the pathways found, validating its critical role. Notably, it's an RNA helicase, which is involved in essential cellular functions like transcription and RNA processing. Its high expression in thalassemia patients, along with network prominence and pathway involvement, made it a compelling therapeutic target. Now, through this multi-layered analysis, DDX3 emerged as a promising and novel therapeutic target. For those unfamiliar, as I said before, DDX3 is an RNA helicase, which is involved in various processes such as transcription, translation, and RNA metabolism. It was highly connected in the interaction network, consistently ranked high across all centrality measures, and appeared in multiple disrupted pathways. Yet, very few studies have actually explored DDX3 in the context of thalassemia, which is what made my study novel. Lastly, our last step was to conduct virtual screening and molecular docking. We now moved into the realm of drug discovery. So using PYRX or Pyrex, we conducted virtual screening of hundreds of chemical compounds from 2 major libraries, Chembil and PubChem, libraries that are well known across the world. This helped us simulate how well these compounds might bind to the DDX3 protein. From this, we shortlisted ligands with the lowest binding affinities, which indicated the strongest interactions. The lowest binding score with DDX3 was an impressive negative 10.2, which was recorded for compound named neotetrazolium, which is from the Chembil database. To take this even one step further, we visualized this ligand-protein interaction in BioVia Discovery Studio. We basically examined how the molecule fit into the DDX3 3D structure and identified amino acid residues involved in the binding. To validate this, we cross-validated DDX3's role. This was done through multiple levels of bioinformatic analysis, as I discussed previously, independent pathway mapping, interaction network strength, and of course, consistent docking results across different libraries. In fact, upon showing this project to an expert, Dr. Sujit Nair, a chief scientific officer in the pharmaceutical industry, he commended my work for being novel and promising for future wet lab validation as well. So, how does my project solve the problem at hand? Currently, Currently, research only focuses on the HPB gene. Mine went to a broader genetic landscape and explored DDX3 as well. Additionally, currently, there's an over reliance on blood transfusion and gelatin therapy. Mine, however, offered molecular target based interventions. Lastly, currently, there is limited drug discovery in this field. However, through bioinformatics, I was able to accelerate screening via computational tools. In the future, this project has a lot of implications. Number one, and most importantly, wet lab testing. The most immediate next step is validating the efficacy of neotetrazolium and the other shortlisted ligands with low binding affinities, in vitro and in vivo. If successful, it could become a real candidate for drug development. Next, protein based therapies. With DDX3 as a protein target, pharmaceutical strategies such as monoclonal antibodies, small molecule inhibitors, or even RNA therapeutics become viable. Lastly, since we used gene expression data, our approach can be adapted for personalized treatment plans, especially relevant in India because we have such a genetically diverse population. Before I end, I want to take a moment to reflect. This project was not just a research challenge, it was a journey of curiosity, resilience, and purpose. As a high school student, I often feel that scientific research is reserved for labs and PhDs. But through this experience, I learned that with the right tools, mentorship, and mindset, I too can contribute meaningfully to solving global health problems. This experience has also now shaped my own aspirations. I can see the vast potential in computational biology as a bridge between data and medicine, and I hope to carry this vision forward in my future academics and professional life. To summarize, I identified DDX3 as a novel and powerful therapeutic target in thalassemia. I used a multi-pronged bioinformatics approach, one that now can be replicated in future studies, from gene expression and analysis to molecular docking. I discovered neotetrazolium as a promising drug candidate, and I laid the foundation for a new direction in affordable protein based therapies that could dramatically improve outcomes for thalassemia patients across the world. Now, one big issue that I did face, or I still am facing, in fact, is that my access to a wet lab was very limited because of the unavailability of neotetrazolium. So this means that I had to rely entirely on computational simulations, which, while powerful, of course, have their limitations. In the future, if possible, I would love to be able to test and validate my research in the wet lab and hopefully contribute to drug development. Thank you so much for your time, your attention, and your belief in the power of young researchers to drive innovation. And thank you so much to CCIR for giving me this opportunity.
Thank you very much, Samaya. It was not just a wonderful and informative presentation, but you made some great remarks and some great comments. Um, this is something that I've faced too, that when you want to contribute to the world of research at a school level, it's not always possible. But I started early, like you did when I was in school, and did some work, secondary research, and then utilize it later when I reached at the master's level. So hopefully you would be able to do that too. But it's great that you're taking initiative and doing all this work, which can actually make a big impact. So congratulations on that. And I just wanted to ask if you have published your research anywhere, like in a
So,
journal or yeah,
yeah,
go ahead.
so it's actually, so I did this research late last year, and I have been in talks with a journal for about 6 to 7 months. And though we finished validating everything, we've submitted all our papers, it still hasn't been published yet. It's in the process of being published, but hopefully soon.
Amazing. That's lovely to know. I hope you're able to publish your research, and then people who are working in the wet labs or in universities can take it from there on. And maybe there would be a better treatment option, which is more accessible, and people know more about an alternative method and a potential gene target. So, great job. And I wish you all the best with everything that the future holds for you. Thank you very much for presenting your work to us. And with that, I will open the floor for any question and answers that people might have. So if you see, there would be a tab saying question and answers on your screen below. So if there are any new questions, those would pop there. So we'll wait for 2 to 3 minutes and see if there are any questions. And in the meantime, you can tell me about behind the scenes of doing all this. Although you have shared a little bit about it, that how you felt and how things were not as accessible. You also talked about India's landscape. So if you want to talk more about that, please feel free to do so.
Sure. So when I started this project, I was actually just entering the 11th grade. So I was quite young, but I did get mentorship from a professor in Delhi, which is the capital of India. And, she really helped me through the process. It took about 6 months, but with her mentorship, it was actually not too difficult to actually do the research computationally. But of course, as I said, still no scope for wet lab analysis, which is one big limitation. Um, but yeah, that was kind of how it went. Yeah.
Amazing. So you mentioned that clearly there are some things that could only be done in wet lab. Can you talk about more about the limitations that come with doing things computationally, and what are the fears that you might have about things just being computational and not having a wet lab component in it?
Of course. So of course there will always be multiple studies done computationally, right? But unless you actually test it in the wet lab in vitro and in vivo, you cannot generalize it and you cannot actually produce it, right, as a drug to humans, of course. So my worry is that this might go to the wet lab and fail, but I would rather know that it has failed, but I've tried, as opposed to just, it being stuck in computational study, right? If I computationally give it to the lab and they start wet lab analysis, it fails, we can go back and refine it. And we can keep doing that till a drug is accurately made, for the general population. But as of right now, it's more like I'm just stuck and I can't go forward.
Got it. Got it. Some great points. You make some great points there. So like you said, things need to go into wet lab. And when things go into wet lab, many a times they're tested on animals. And then after that type of trials are passed, then we progress to testing it on patients, right? So there's several stages which come with any testing of any type of genes or any type of drug, or let's say. So there have been many studies going on, right? If we would be able to eliminate animal studies completely, do you think that we would be able to reach that given that computational power is getting so strong and we are progressing so much in the field of AI or LLM? So what are your thoughts on that?
So of course, for years and years, we've been testing on animals first. Um, for most diseases, unless of course it's like cancer and things that where humans have to be tested on first. But with even with computational biology and computation, any type of computational science, I'm not 100% sure that we'd be able to completely eliminate animals, though of course in an ideal world it would be the most ideal thing to do would be to go straight to patients and like knowing that it is going to work. um but just to keep patients safe and to keep humans safe first, I don't see animals being removed from the testing process for a while. I don't think AI is ready for it yet.
Thank you very much. Thanks for sharing your thoughts. And I don't think we have any open questions at the moment, but it was lovely seeing your presentation and hearing your thoughts and ideas about the world of computation.
Thank you so much for having me.